Welcome to the third video of MBBS studies, and in this video I will be going to explain about the rest of the topics of cell, and this will be the cell part 2 of the video. Here I will be explaining about the plasma membrane. Here each cell is covered by a membrane called the cell membrane or the plasma membrane, which separates the cell from the external environment. It is about 8 to 10 nanometer in thickness. Structure of cell membrane. It is described by fluid mosaic model. S.J. Singer and G.L. Nicholson proposed the fluid mosaic model in 1972 to explain the structural composition of cell membrane according to the fluid mosaic model. The cell membrane is made up of bilayers of phospholipids and small amounts of glycolipids and cholesterol along the protein and carbohydrates. Next, the lipids. The phospholipid is the principal lipid present in the cell membrane. Small amount of glycoproteins and cholesterol are also present. All the membrane lipids are amphipathic in nature as they contain both hydrophobic and hydrophilic regions. Next is the amphipathic lipids are arranged with their polar head groups oriented towards the extracellular and the cytoplasmic side, and the non-polar hydrophobic tails oriented towards each other in the direction of the hydrophobic core of bilayer. Proteins. The proteins present in the membrane are categorized in two types: peripheral proteins and integral proteins. The peripheral proteins. These proteins are loosely attached to the outer and the inner surface of the membrane by weak ionic and polar bonds. They can be easily removed without disrupting the membrane. Integral proteins. These proteins are firmly embedded in the bilayer and tightly bound to the lipid bilayer by hydrophobic bonds. They cannot be removed without some of these integral proteins span the whole bilayer and are called transmembrane proteins. Transmembrane proteins serve as transport or carrier proteins. Carbohydrates. The carbohydrates exist as glycolipids and glycoproteins. They are always found attached to the external surface of the membrane is called glycocalyx. And there is now the function of the cell membrane. There is a mnemonic for the function, that is P2SE2. If you remember this mnemonic, then you can easily tell the function of the cell membrane. First, for P2, that is provision of the shape and size of the cell, and protection and support. For this, P2. And for S, that is selective permeability. For E2, there is excretory function and exchange of gases. Now, at first, provision of shape and size of the cell and size to the cell. Cell membrane maintains a definite shape and size of the cell. Protection and support. The cell membrane acts as a barrier between the cell's internal environment and the external environment. It regulates the passage of substances in and out of the cell. Selective permeability. The cell membrane is selectively permeable, meaning it allows certain substances to enter or exit the cell without restricting the movement of others. Excretory function. Waste products are thrown out of the cell through the cell membranes. And exchange of gases. Exchange of gases takes place through the cell membrane. These are the 5 functions of the cell membrane, and we can remember it through the mnemonic P2SE2. Now, the endoplasmic reticulum structure. It is the next topic. The endoplasmic reticulum. This is a membrane channel system connecting the nuclear membrane and the plasma membrane. They have network-like system types. There are two types of endoplasmic reticulum, namely the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Smooth endoplasmic reticulum has smooth appearance which has no attached ribosomes, and rough endoplasmic reticulum has rough appearance because of the presence of ribosomes on their structure. Now we will be telling about the endoplasmic reticulum functions. In the functions, there includes mechanical support and lipid synthesis and protein synthesis, then detoxification, reservoir of calcium, and protein secretion. For it, we can say about the mnemonic, that is MLPDRP. We can say we can tell a mnemonic for this function, that is MLPDRP. This is a mnemonic for the functions of this endoplasmic reticulum, that is mechanical support. Endoplasmic reticulum gives mechanical support to the cell. Endoplasmic reticulum gives mechanical support to the cell, and in the lipid synthesis, endoplasmic reticulum is responsible for the synthesis of lipids such as triglycerol, cholesterol, and phospholipids. In protein synthesis, rough endoplasmic reticulum helps in protein synthesis as they have ribosomes. In detoxification, steroid hydrolyzation, detoxification of certain drugs as they take place in endoplasmic reticulum. And in reservoir of calcium, endoplasmic reticulum acts as intracellular reservoir of calcium. In protein secretion, endoplasmic reticulum also helps in transport of synthetic products like glycoproteins and lipoproteins out of the cell. Now, our next topic is subcellular fractioning of cell organelles. It includes nucleus. It contains the genetic material, that is DNA. DNA replication and transcription occurs in the nucleus. Now, the next topic is endoplasmic reticulum. It is the marker enzyme for endoplasmic reticulum is glucose 6 phosphatase. The membrane enclosed spaces are the endoplasmic reticulum. There are two types, smooth and rough endoplasmic reticulum. Synthesis of proteins, glycoproteins and lipoproteins takes place in endoplasmic reticulum, and also the metabolism of drugs. And in lysosomes, it is a bag of digestive enzymes of the cell. Degradation of proteins, carbohydrates, lipids occurs in the lysosomes. In mitochondria, it is the powerhouse of the cell. ETC, that is electron transport chain, ATP synthesis, TCA cycle, tricarboxylic acid cycle, fatty acid oxidation, ketone body production occurs in mitochondria. Part of heme synthesis, urea cycle, glyconeogenesis, pyrimidine synthesis occurs in mitochondria. In Golgi bodies, we can say it is a network of latent membranes and vesicles involved in protein packing and storing secretion. In peroxisomes, we can say contains peroxides and catalases, and oxidation of long chain fatty acid occurs in peroxisomes. In ribosomes, these are the factories of protein synthesis which are found on the endoplasmic reticulum. Cytosol, cytosol has gel-like matrix surrounded by cell membrane. It is a site for protein synthesis, glycolysis, glycogen metabolism, and fatty acid synthesis. Thank you.